Hi, this is Arijit from Adamas University, Kolkata, India, and I am going to offer you a glimpse of one of our project, which is on Leucine Carboxyl Methyltransferase from Leishmania and deciphering its interactome. So the project originated from a genome-wide gain-of-function analysis against a drug called sinefangine, which is a methyltransferase inhibitor, and this screen led to identification of several genes, including leucine carboxyl methyltransferase, which is involved in resistance against this particular drug. What this enzyme does, this enzyme transfers methyl group to the C-terminus carboxyl group of any polypeptide that encodes leucine. So these leucine containing polypeptides are methylated by this enzyme, and this methylation leads to regulation of the enzymatic activity or other function of these polypeptides or these proteins. Now, when we analyze this LCMT protein from Leishmania infantum in terms of phylogeny, we found out that all the kinetoplastida orthologs of this gene clustered together, and this cluster is pretty distant from the cluster that are obtained from the eukaryotic orthologs. So they are relatively phylogenetically distant. And despite of their phylogenetic distance, we found out by multiple sequence alignment that the key residues important for catalysis, they are conserved across domain. Now, to be really important for the biology of Leishmania, it should have sufficient substrate in the proteome of Leishmania. And when we analyzed almost 8,000 proteins in Leishmania, we found out that more than 10% of those 8,000 proteins contain a C-terminal leucine. So all of these proteins can have a possibility of being a substrate of this enzyme, and therefore this enzyme might play a very crucial role in the biology of Leishmania. In order to decipher its role, we make knockout of this enzyme in Leishmania infantum, and these knocked out cells were little bit sluggish in terms of the growth rate. These cells showed enough resistance against sinefangine, these knockout cells. Interestingly, the overexpressing cells also showed resistance, but the overexpressing cells showed maximal resistance at early log phase of growth of the parasite. However, this resistance depleted with the mid-log and late log and stationary phase. Whereas for the knockout strain, this resistance level was maintained throughout the life cycle stages. So this means that because the resistance level is stage dependent, that clearly indicate that this LCMT enzyme has significant role in terms of regulating the biological responses processes in this parasite. Now we built up a homology model for this enzyme from Leishmania, and this model uh, satisfied all the normal criteria for evaluating homology models. And to test whether this enzyme is really a good methyltransferase, we tested several methyltransferase inhibitors. We docked this inhibitor against this homology model and found out that the common methyltransferase inhibitors, including formycin, azadenosine, or desaneflanosine, all of these inhibitors docked pretty well with the active site of this enzyme. So this clearly indicates that this leucine carboxyl methyltransferase is a potentially active enzyme in Leishmania. Now, in order to decipher its interactome, we expressed H-attacked version of LCMT in LCMT knocked out cell. We immunoprecipitated that with anti-HA antibody and analyzed the immunoprecipitated fraction with LC-MSMS. And from that analysis, the peptides that we identified, we further analyzed those peptides through a pipeline called SAINT, which is a probabilistic pipeline for identifying potential interactome after obtaining LC-MSMS data, and thereby we identified a candidate interactome for LCMT from this parasite. And this candidate interactome includes 411 proteins, and interestingly, out of these 411 proteins, 46 proteins harbors leucine at its C-termini. So these red dots are those 46 proteins that harbor leucine at its C-termini, and when we analyzed Go enrichment analysis of these 46 proteins, we found out that one of the most potent term that was enriched is translation factor activity. And this translation factor activity involves some ribosomal proteins, some initiation factors, some elongation factors. So there are a lot of genes that are involved in translation process. Now we screened all of those genes for in silico protein-protein docking with L-infantime, and we found out that at least some genes, 6-7 genes, they can potentially interact with LCMT. with satisfactory del G value, which indicates the level of interaction, and sufficient area interface for protein-protein interaction. So this is the surface area of interface. So all of these proteins that include some elongation factors, some ribosomal proteins, some small RNP protein. So all of these proteins interacted pretty well with LCMT as reflected by our in silico protein-protein interaction method. And one of the candidate interaction, which is of particular interest, is that of elongation factor 2 beta protein with LCMT, and we are presently working on it. So to summarize, so far what we have worked on is deciphering the interactome of LCMT and in silico analysis of candidate interacting proteins with LCMT. In future, we would like to focus on the ribosomal proteins and elongation factors that are interacting with LCMT, and we would like to further decipher the regulatory role of LCMT on ribosomal action. So that is our future target. This is the model that we think that sinefungin, the inhibitor, it inhibits mRNA capping, and because LCMT can contribute to resistance against sinefungin, it can potentially regulate the process of translation initiation, and we want to address this gray area which is not yet clear. So I would like to acknowledge Professor Mark Poulet and Philippe Leprohan of University Laval, Quebec City, Canada, with whom I am in active collaboration. I would like to thank Akash Mitra, who is working with me on this project. And I would like to thank all the open source software providers. And finally, I would like to thank Adamas University for hosting me and for allowing me to perform this work. Lastly, thank you all for your attention and interest in our project. Thank you all.